Hello, everyone, and thanks for joining today's Exchange Seminar Series. So our today's speaker is Dr. Rhee, and this is my pleasure to introduce you to everyone. So Sue received her bachelor degree in biology from Swarthmore College in 1992, and then a PhD in biological science from Stanford University in 1998. Then she joined the Carnegie Institution for Science in 1994 as a research associate, and then promoted to staff associate and staff scientist in the same place. And in 2016, she acted as the acting director in Department of Plant Biology, Carnegie Institution for Science. And currently she's in Carnegie Institution as a senior staff scientist. So she's been actively involved in several scientific advisory boards, grant review boards, including DOE-related boards and international conference organization boards. Sue has been contributing to sciences in very active and influential ways. So, for example, she acted as founding director for the Arabidopsis Information Resource, or TARE, while she was an early career investigator at Carnegie. For that purpose, she led a team of biologists and software engineers to create a computational infrastructure called TARE, or Arabidopsis Information Resource, to collect and encode all available genomic and literature data to be computable by algorithms and easily accessible by researchers. So TARE has been a significantly important platform and a revolutionary database for plant research by enabling systematic and quantitative analysis of biological functions and pathways. So thousands of scientists around the world, including myself, are still actively using this platform. So in addition, her group was one of the early developers of the gene ontology system, where she and her team contributed to making the system to work for plant genomes. And as you probably know, gene ontology has been influential in analyzing and interpreting genome and post-genomic data across many organisms, and have been used to analyze data in thousands of research articles, including many studies of various human diseases. She's been also leading projects aiming to systematically infer functions of genes and group them into pathways, annotate enzyme function on the genome scale, and establish a unique and unified resource of plant metabolic networks. For example, PlantPsych is one of the important resources for scientific community that provides access to manually curated or reviewed information about shared and unique metabolic pathways present in over 500 plant species. So her very recent activity and contribution to science is leading efforts on developing Plant Cell Atlas, or PCA, which we will hear more about it today. Thank you so much, Sue, for accepting our invitation. This is very exciting to have you here. So this is all yours now.
Thank you so much, Amir, for that very generous introduction and for the invitation to speak at EMSL. It's a real pleasure to be here today to speak with you all about the Plant Cell Atlas initiative. I'm sorry that I'm not there in person, but I'm really hoping that I can come visit sometime soon. So I'd like to start by sharing with you some of the most stunning images of plant cells that are generated by the Plant Cell Atlas community here. And as Amir mentioned, I'm one of almost 50 people who are trying to get started with this initiative called the Plant Cell Atlas. And as you might see up here with your Amir and Chris Enderton at EMSL having played prominent roles in the development of this initiative from the beginning. So this set of core members of the PCA initiative is very diverse, almost 50% women and 12% otherwise underrepresented, and almost 70% almost 80% early career scientists, in fact. Oops. Did I just unshared?
No, we still see your slides.
You do, okay. Oh, okay, good. Sorry about that. And so these core members come from institutions worldwide, including public, universities and colleges, as well as nonprofit organization and even government institutions like PNLL. So as plant scientists, we care deeply about food security, environmental protection, and energy production under the conditions of this looming climate change. And this is actually one of the things that concern me the most these days, in that biomass is actually going to become one of the major sources of primary energy in the next 30 years or so, as shown on the graph on the left. You can see that the red bars represent the projected amount of primary energy coming from each of these sources by 2050, and biomass, as you can see on the far left, really constitutes the most amount of energy source. And that's really good for plant scientists and plant biology in general, but biomass is actually a really huge energy hog, and has a huge amount of land use. And so, I think we have a lot of challenges ahead of us to try to figure out ways of producing bioenergy in ways that are sustainable. And so I think one of the most important and interesting questions that we face in plant science in the next 10 to 20 years is really the question of how can we get the most out of plants, such as energy, food, materials, medicine, so on, while minimizing resources and reducing environmental impact. And for that, I believe that we need to be able to develop programmable plants. And in order to do that, we need to really understand deeply the major processes that go on in plants, such as development, reproduction, metabolism, defense, and so on, and how they're connected to each other, how they work together at molecular to ecosystem levels. And so, when thinking about how we can make the most impact in science and society today, especially with an emphasis in plant biology, I think that we need to really figure out a way of connecting the fundamental discovery to translation, to using those translations into actual solutions and putting those solutions into action. This whole nexus of from discovery to action has to be accelerated. And I think, though, we still have a pretty big bottleneck on the front end here with discovery and translation. And so that is really the subject of the Plant Cell Atlas Initiative. In order to really accelerate scientific progress, especially in plant sciences, I think there are two major bottlenecks that we have to overcome. One is that plant science is still quite underrepresented in STEM. So this is a graph that shows the number of soil and plant scientists working in the US today relative to the number of medical scientists, and it's only about 10%. So we do need to still bring in a lot of talent into plant science. A second challenge that I think we face is that we still really don't understand how most of the genes function and gene products function. And this is a graph that's generated from gene ontology annotations that we just put together this week, in fact. And we actually wrote, Bo Shui, who's an RA in my lab, wrote codes to actually generate this on a regular basis now. And we'll actually try to do this for the major plant species. But these are some of the most heavily studied model organisms today. And they show in each pie chart the relative proportions of genes and gene products that have been characterized. And the green pieces represent those genes that have been characterized experimentally. And the blue pie pieces of the pies represent the genes that have been characterized just based on sequence similarity, and it's just predicted for function. And the black pieces represent those genes that are completely uncharacterized. In other words, we don't even know what they look like. They don't look like anything that's been characterized experimentally. And so besides yeast, which actually has very small pieces of the black, slices, most organisms that have been intensively studied still have pretty big portions of the genomes that are completely uncharacterized. So I think this is actually one of the major hurdles of really trying to understand and engineer organisms. And so how can we accelerate, that kind of fundamental discovery of gene function at a sort of systems level and global scale? And to talk about that, I like to use jigsaw puzzle as an analogy. And so, when you start doing the jigsaw puzzle, at first it's actually kind of hard because all the pieces are sort of without context and difficult to place. But as soon as you start to put some pieces together and some areas become a little bit more visible, then it really is much easier to put the rest of the pieces together, and the process of putting together the whole puzzle really accelerates. And I think we could use this analogy to biology too. And so then, um, we might think about what are those contextual anchor points that can be used to really accelerate the discovery of gene function. And I think one such obvious context mapping sort of anchors is really the location of the genes and gene products within the organisms at different scales. So, many of you are probably aware of the sequence to function paradigm, where we can try to infer function based on sequence and structure. Um, but location to function paradigm is really kind of not as, I guess, well explored or exploited compared to sequence to function paradigm. And so, within the Plant Cell Atlas Initiative community, we are trying to think about ways in which locations of proteins and other gene products can be used to infer their functions. And we think this can be done in a systematic and efficient way from many scales, ranging from molecular to cellular to tissue, organ, and organism levels. And more information about this has been written up in a roadmap paper by the consortium, and it was just very recently accepted in eLive. So I think it will appear online in the next few weeks. So in order to really realize that location to function paradigm and to create a community around this concept, we initiated a grassroots effort called the Plant Cell Atlas a few years ago, whose goal is to map all of the components of plant cells from nano to macroscale resolutions. And there are a few papers that have already been written up about this initiative. And to sort of describe a little bit more the goals of the PCA initiative, primarily we want to identify the cell types, states, and transitions of plant cells in plants. And we also want to map proteins to cell types and subcellular structures. We want to also be able to track the dynamic interactions among those proteins and integrate these data to generate multi-scale models of cells and tissues. We think that this initiative will have a large impact in a few different ways. First, data and tools coming out of this initiative should be able to answer fundamental questions in plant science. We also think that the data and resources coming out from this initiative could accelerate plant engineering for agriculture, bioenergy industries, as well as managing the ecosystems. And finally, we think that there may be a lot of cool things that we could engage the general public with that will be coming out of this initiative. So we think of this Plant Cell Atlas research framework in the following way. We think this has to be a very close collaboration among three sort of areas. One is plant science, and second is data science, and third is technology development. And yeah, I know that EMSL is really one of the frontiers in technology development and application, as well as data science and some plant science as well. And so I think that places like EMSL have really big roles in making this PCA initiative flourish. So now I will tell you a little bit about the kinds of plant science that could be enabled by PCA research framework, and then some of the emerging technologies that might be needed to achieve the goals that we have laid out. So to start with, you might be wondering what kind of science PCA might be able to enable. And I think, it will actually impact all major nodes of plant biology, really, ranging from genetics to cell biology to development to metabolism and modeling and so on. I'm not going to really go through all of the areas that the data and tools from Plant Cell Initiative would influence, but I'll give you a couple of examples. The first example I want to talk about is C4 photosynthesis. Um, which is really an amazing machinery and process done by all plants. So plants in the ocean and the land can actually capture about 210 gigatons of carbon dioxide per year. That's almost 20 times the amount of CO2 emissions that humans make. A lot of this carbon captured, of course, is really re-released by respiration of plants and microbes, of course. But the point is that plants are capable of sequestering a huge amount of CO2. And what's even more amazing is what's called C4 photosynthesis, which is a way to concentrate carbon dioxide. And it's only been invented by about 3% of flowering plants. But those 3% of flowering plants are responsible for capturing about a quarter of carbon dioxide in the atmosphere today. So it is an extremely efficient way of fixing carbon. And just to very briefly summarize, in most plants that do photosynthesis, they do C3 photosynthesis, which is basically capturing CO2 into a 3-carbon sugar. And they do it in these cells called mesophyll cells in the leaf. But in C4 plants, that photosynthesis process has been separated into two cell types that really do division of labor in terms of carbon fixation from carbon capture. And so this cell type separation and innovation really enable this amazing feat in photosynthesis. But we still don't really understand all of the ways in which these two cell types develop and function, and how they respond to different environmental conditions. And so I think single cell biology could have a huge impact in allowing us to understand how these two cell types work independently and together with each other. And not only that, chloroplasts themselves are really amazing organelles that have an extraordinary diversity. Not only diversity in their structures across plant lineage is shown here on the upper left panel. They can be really different in terms of locations within the cell. So in land plants, most chloroplasts are found in cells in the inside tissue of leaves, although in aquatic plants, the chloroplasts are most concentrated on the epidermal cells. So that variation and diversity is really interesting adaptation in environment, and we don't really know how that's done. And lights can change, and the quality and intensity of lights can change at millisecond levels, maybe even microsecond levels during the day. And chloroplasts can move around within the cell to actually maximize light capture, as shown in the lower left panel here. And we have multiple types of plastids that have different functions, not just chloroplasts, but we have chromoplasts in senescing tissues and leucoplasts and roots. And so, there is a really rich set of biology in studying these amazing organelle, the chloroplasts. And again, I think single cell biology and plant cell initiative could have a huge impact on uncovering a lot of these amazing things and their mechanisms. The second example I would like to give on how the PCA initiative can have a big impact on is plant-plant pathogen interactions. So each year, about 20 to 40% of crop loss comes from infections and diseases caused by pests. And that is about $220 billion a year globally. And there's a lot that we know about plant pathogen interactions, and a lot of engineering approaches have been implemented already, but there's still a lot we don't know. For example, we don't really know exactly what the nature of the infection processes at the individual cell level and how that changes temporally and spatially. So, those are just a couple of examples of cool science that I think the Plant Cell Initiative could really open new doors in. But there's many, many more examples, of course. And so in order to enable all these cool science, like what are the technologies that we need? And here I attempted to list just a few that I could think of and things that have been discussed in the initiative amongst members. And they include things like single cell sequencing, high content, high throughput imaging, single cell proteomics, spatial proteomics, of course, automated annotation using AI, high performance computing, visualizing data, and integrating different types of technologies. And I think this is actually, an area where EMSL really excels in. And so I am really looking forward to, having more conversations about this with you, either after this talk or later this week. I would love to learn more about the kinds of technologies and capabilities that you have, and how what you can do can be useful for really moving this initiative forward. Just giving a couple of examples of the power of technologies that can be useful for the PCA. One is, of course, the single cell RNAseg technology. And this is taken from a really nice paper that Ben Cole and Diane Dickel and all have published recently in Communications Biology, based on a workshop that they organized. And this figure just shows you that compared to the bulk RNA-seq analysis on the top row, higher and higher resolutions and specificity of being able to probe transcriptome data at single cell level will really increase the amount of knowledge we can gain, the kinds of questions we could ask. In this case, as for an example, for plant pathogen interactions. Another really cool thing that came out of that workshop that's available from the workshop report is this diagram from Anup Sinj that describes an example of a microfluidic platform for integrating technologies such as live cell imaging along with immunohistochemistry and maybe doing proteomics and so on. And so I think this is, a really cool idea of technology that I think could be hopefully adapted to plant cells. And with all of these data and tools, we really need to also think about developing a one-stop shop of the data. And, we imagine having, a nice and easy access to all of the data that's connected to each other. And it would be really interesting to have conversations about how this type of infrastructure could materialize. And so if we were to able to generate those data, make those tools, and have them available, what are some of the opportunities in bioproducts and biomaterials? And so I'll give you a few examples, again, from the workshop that Ben and Diane organized. And so this first one is really identifying regulatory elements for engineering purposes. So let's say that you're really interested in trying to produce a certain plant chemical, and you could first find out which cell types that chemical is made, and then by single cell sequencing, and then getting all those genes that are co-expressed, and then looking for the elements that are responsible for that specificity of expression of those genes, and then taking that sort of regulatory elements and expressing it on genes that you want to express specifically in that cell type. Similarly, you could imagine using single cell profiling of the transcripts and proteins to find metabolic pathways that might be actually co-regulated and be expressed to make compounds of interest. And so this could be really interesting and useful for pathway discovery. And finally, the single cell technologies might be really useful in performing high throughput phenotyping. Phenotypes of interest, such as like making a lot of oil. We could try to think about doing this in eukaryotic, single cell eukaryotic systems like CLAMI, or even protoplasts or other types of cell culture systems. And if we are able to phenotype at an individual cell level, then we may be able to really expedite the discovery of phenotypes or traits of interest for engineering purposes. Okay, so I'd like to start wrapping up by talking about what are some of the challenges and opportunities that are facing us and the PCA initiative. There are several of those that are articulated in this roadmap paper in eLife. But just to summarize some, we think that the ability to expand the single cell omics technologies to multiple plants so that we can do comparisons and translational biology would be a really important thing to accomplish and overcome. And we also think that integrating different types of omics technologies into the same cell types would be really important. And other things include, being able to annotate the plant cell types using markers that could really ground us in analyzing all sorts of single cell profiling data in consistent ways. So I'd like to just describe how we're trying to go about continuing this community building and implementing some of the things that I described to you today. So in the Plant Cell Atlas Initiative core group, we have those about 50 people sort of working in committees. We have about 14 committees listed here, ranging from different types of scientific areas, as well as things like data management and data infrastructure and communications to the outside. And those committees are led by this set of really amazing scientists, very diverse, 45% women, and mostly early career scientists. And I want to actually shout out to Dr. Selena Rice, who is the coordinator for the PCA initiative and who's really been doing the lion's share of the work behind the scenes. And the initiative is also supported by a set of a steering committee, including myself, and Diane Dinkel at LBL, David Earhart at Carnegie, Ken Birnbaum at NYU, Marisa Otegi at Wisconsin Madison, and Nick Provart at Toronto. And together we have made some projected milestones. We are in the very beginning of this milestone down here in 2021. And this set of milestones is sort of color-coded in 4 areas: data generation, data analysis, software development, and community building. And so, as I've described to you, we've started building the community, and it's going very well, actually. And not a whole lot has been done in terms of building software, although I think there's a lot out there available from the Human Cell Atlas Initiative that we should leverage. Data analysis and data generation have already started, of course, outside of the PCA initiative. And a major goal for all of us in the community is really how to try to bring together these types of data that's being published, in many places, and try to sort of be able to compare them across experiments and studies and species and so on. And so I just want to highlight some of the things we have achieved in the last couple of years. So in 2019, we had a first vision paper. We ran a small NSF workshop grant. So we ran a small NSF workshop funded by NSF. And then we launched the website and Twitter. And then in 2020, we had, so in 2019, we wrote NSF, we wrote a grant to NSF to do a workshop. And in 2020, we did the workshop. This was actually done virtually because of COVID. But because of that, I think we ended up attracting a lot more people than originally intended. I think we originally intended to have about 30 people on site, and we ended up doing it virtually, and it brought in about 400 people. And then we also had a call for papers for a focus issue on plant cell atlas in plant physiology. That's actually going really well. It's gonna come out in January, February of 2022. And then we also had published the report on the inaugural workshop in Plant Direct. The first half of 2021, we launched a YouTube channel, and we then held a single cell sequencing workshop, again virtually, which was also well attended, and then spatial proteomics workshop in April. We actually started a research collaboration network grant, again funded by NSF. This will go on for the next 5 years. We also submitted an application for a Gordon conference on PCA that will start, if funded, that will start in 2023. And then we just had this roadmap paper accepted at eLive. And we have a bunch of other things in progress. So we are going to be launching the PCA website, second generation of the PCA website in the fall. And we are actually doing a art and science exhibit at 3 historically Black colleges and universities in September. And then we also wrote a short paper describing that exhibit, which will come out of Trends in Plant Science in September as well. And the training committee is actually going to set up a writing accountability groups for PCA community to help each other on learning how to write grants and papers. And then Chris Enderton at EMSL is actually organizing a really nice mass spec imaging workshop in October. And that's been announced recently, and I hope you could all make it there. And then finally, we're going to have an international symposium on PCA in December of this year. And it will be taking place over 2 days, again, virtually. And I'm hoping that we can attract a lot of people to that. We have a lot of stakeholders in this community, besides the Plant Cell Atlas community of scientists and engineers and so on. We also really want to bring in people from a lot of other sectors, such as industry and breeding and undergrad and general public sectors. Currently, we have about 800 members in our email list, and we're followed by about 600 followers in Twitter, and our YouTube that was launched recently has 60 or so subscribers so far. So if you're interested in getting involved in this activity, you can find out more information about PCA on our project website called plantcellatlas.org. You could also join the email group by going to this contact page and just putting in your email address and saying that you want to be part of the PCA email group. You can follow us on Twitter. You can go to the YouTube channel and check out our videos and subscribe to it. or email us at this email address, plants.atlas@gmail.com, if you have any other questions. So that is the end of my talk. Thank you very much. I'd be happy to take any questions if you have any.
Thank you so much, Stu. That was a great talk. Really appreciate it. So are there any questions?
Hello, hello, Sue. Can you hear me?
Yeah.
Yeah. Hi. It was a good talk. So I attended the symposium as well. So I just have this question, like the data is going to come from different labs in different places of the world. So there is going to be what are the challenges that will be there to handle such data sets? So I mean, there will be some technical variations that will be in the data, right? Not actually representing the biological variations. So what will be the kind of challenges and the PCA team, how they are preparing to deal with such kind of data that is coming from wide different labs and different places?
Yeah, I think that's a really good question. And I think that will be a really big challenge for anyone who's interested in analyzing data, that's been published by other groups, or, incorporating other people's data into their studies. And, we've of course experienced this even before the PCA initiative with microarrays and RNAsegs and so on, proteomics data to a lesser extent. So it's not necessarily a new problem, but I think it's something that hopefully a community like this could sort of work together in overcoming faster. So, certainly things like having benchmark sets and reference sets and protocols and how to sort of, analyze the raw data in a consistent way, but also maybe trying to come together and mitigate the problem earlier on by just maybe coming up with best practices and standards in how to design experiments and what are the really important parameters that should be captured when performing an experiment and reporting the results. So those kinds of standards, hopefully we could, come together and agree upon, and then sort of either publish or make available online so that people will take a look at that even before, generating the data, which would be more beneficial, I think, than trying to figure that out after the fact.
Right. Great.
And
Thanks.
so I would say, yeah, Ben Cole is the lead for this data management committee, and they are actually putting together a set of papers that have generated a lot of nice single cell data, especially those that have been done in ways that are easier to compare across studies. So they're going to actually put together a list of those kind of benchmark sets or recommended sets for people to use, and we're going to release that online when we update when we do the website launch in the fall.
Okay, great. Thanks.
Thank you. Before we moving forward to next questions, so can I ask you if you probably turn on your video, or actually if you turn off your slide presentation mode, because of that technical issue, we cannot see you.
Okay.
But now,
okay, let me see. Stop sharing. Okay, so now you can see me. Yes.
Not yet.
Oh, really? I can see.
Well, I can see you, Sue.
Yeah, we
I can
can.
see you.
Oh, but I can't for some reason. Anyway, but that's okay. So we have, so Allison, you have a question. Go ahead.
Oh, yeah. Following up with Vimal's question, what are some of the biggest challenges and opportunities you see in data integration?
Data integration, some of the challenges, I think that, things that I just discussed earlier, being able to compare data across different studies, and some of the opportunities, I think that, like, ways to be able to capture different molecules from the same cells would be really amazing. So that we can actually look at metabolites and proteins, from the cells that have been treated the same way. I don't know. Like, what do you think? What is your sort of view of the major challenges?
No, I think you hit the nail on the head in being able to utilize data from multiple different like universities or instruments or laboratories. I think another challenge can be the scale of different data sets and the databases that we're mapping back to. If we can identify all of the proteins in a system, but only 10% of the lipids or something. Being
Yeah.
able to sort of rectify the disparities in the confidence and the amount of data we can get, I think will be really important.
Yeah. Agreed.
I think one of the things to keep in mind is that if you're building an atlas, right, you can always start with the healthy tissue. And in theory, everybody should have a control there, right? So that's where, instead of focusing on disease states or any kind of change in, the whatever you're looking at the perturbation, just focusing on control across experiments, and build the atlas from there.
Yeah, and especially important would be standardizing nomenclature and ontologies and all of that fun stuff that people don't like to think about, but is really important.
Yeah.
Great.
Yeah, I think this is important to be able to figure out a way to, just speaking of, like, recruiting talent. I think that it's really an important problem. I think there's a lot of value in creating these standards, and like putting in energy and money into making sure that they are used. And this is kind of not, I don't know. I think they're really under recognized, I would say, right? And it's, yeah, and at many levels. And so how to make that kind of stuff fun and exciting is a major challenge.
Great. Move, do you have a question?
Yeah, hi, Sue. Very inspiring talk. I don't know if you saw Jay's comment too. I was kind of relate to your question about the talent, and it was nice to see on the beginning of your slides. It's nice to see those data, quantifiable data, to kind of see how the field compares with the biomedical. And I have a story like that. I was trying to help a collaborator, like plant biologist, hire a postdoc, and then I found some great candidates. And then when I talked with them, they said, no, we're not going to do plant stuff. We want to do like biomedical. So I think it's a really big challenge, I think, and maybe like related to like the funding structure. And I don't know, like I guess because I saw like you have like an industry on board. I don't know like if there's any practice you are thinking about to kind of at least for like a fresh graduates, they're probably like looking more into like what career they can find right after graduation. So if there's some inspiration for them to have better connection to industry, that might help recruit more talents to join the plant science.
Yeah. I think that industry is a missing link for the PCA right now. And yeah, it would be interesting to see if we can figure out ways of connecting with them. But plant industry is also kind of set up in ways that's just like a handful of really big ones, right? And then there aren't that many, a lot of the kind of startups. So yeah, I don't know how we can get involved with the industry sector, but that's something that's also homework for the community, I think. And in terms of attracting grad students, I think a lot of students are really excited about or get motivated about climate change. And so I think that, yeah, I don't know. I think that that we need to figure out better ways of connecting to that, in terms of plant biology. It's very strange. A lot of people talk about sustainability, but they don't really include plant science as an inherent component of that. So I don't understand where that's coming from.
Maybe that's where we start at the undergraduate level, with the idea that folks go into graduate school for this.
Yeah, I think
And
so.
I've seen like some professors, they have like funds from like those like maybe like industry based or some kind of nonprofit. I don't know if there's a way kind of to reach out through those professors at universities. Yeah, I think Jay is making some similar comment. I don't know, Amir, if you see Jay's comment in the chat.
Yeah, I just wanted to mention that. Yeah, Jay has a question. Just mentioned it's about the relative sizes of the biomedical and plant biology fields, and he's asking, are there key pieces of know-how or infrastructure strategies that can be leveraged from the big agriculture biotechnology industry? So Jay, did I communicate it well, or you wanna?
Yeah, I don't know, Jay. I haven't really. I have very little experience with how things work in industry, so I don't know if I could say anything useful here. I do know that there's a lot of effort going into Human Cell Atlas initiative, and a lot of money is coming both from public and private sectors for that. And they have been building really useful tools, which I think we should leverage. And that's the other thing too. I think that, might be interesting to also figure out ways of engaging the private sector, like, philanthropy sector. But yeah, I think.
Thank you.
Much smaller. But in terms of the stuff that the big ag has done, I don't know. What are some of the things that the big ag has done that we can leverage?
I honestly don't know. I happened to do a stint in pharma before coming back to the academic world. And it was just one of the striking things was that when we interviewed folks who were coming out of big ag and looking to just change industries, some of the ways that they worked were incredibly sophisticated, even compared to some of the large investments that were being made in pharma. And so there was clearly some kind of a big gap, even in that private sector, between industries as far as capabilities and approaches. So it just struck me as sort of EMSL in some respects brings some very infrastructural centered development approaches to what we do, and would be great to know more. Especially, I sit in the computing side, and their sort of integration between different kinds of experiments and how we handle our data and metadata is really important to be able to draw deep, broad conclusions. I didn't have any particular ideas in mind. I was just really curious because I appreciated your drawing that contrast between the sizes of the fields. Thank you for the fantastic talk.
Okay, well, thank you, Jay.
Great. So I don't see any raised hands here, or probably there's no more questions.
I sort of had a comment slash
discussion
point, since both Jay's comments and just this discussion, it makes me wonder about specifically genotype phenotype associations. I mean, that's something that I think that big agriculture is leaning on. Like that's what they're trying to accomplish, is they want specific traits in plants that are being grown and trying to capture some of the phenotypic variation, whether it's from engineered plants or wild type plants, and being able to tie those specifically to cellular traits as opposed to macro traits is something that might be leverageable across both research and industry. And I wonder if there might be a way that, you know, whether it's EMSL or some kind of like this also occurred to me on the level of if you were doing or integrating data across lots of labs, it might be really helpful to have a baseline data set that addresses the same exact way across many genotypes of the same plant, instead of focusing on lots of different technological advances that can be done in smaller labs, but something like EMSL or a larger institution that's capable of supporting a greater number of genotypes to do that seems like it would be really powerful for integration.
Yeah. I really like the way you articulated the linkage between cellular traits to macro traits, what I kind of think about as physiological traits. And, you know, we talk about biomass, things like biomass a lot and growth parameters a lot. And, but in like C4 photosynthesis example I gave, you know, we have very little in terms of our ability to really probe things in the two cell types, you know, separately, like in terms of enzyme assays or omics. You know, people have basically tried to physically macerate the leaf tissues to get the mesophils out and then, you know, use the remaining bundle sheet. But even coming from really top C4 photosynthesis labs, if you look at the papers, you know, the method is very crude and you always get contamination, right? And so the resolution and specificity that you get is really poor, I think. And you just think about like the importance of bundle sheath and mesophyll cells and their functions. And we don't have a very sophisticated way of probing them separately. And just solving that problem, I think, could be really transformational. And, you know, we already know that, you know, C4 photosynthesis can be manipulated to a really great extent and has huge potential in affecting biomass traits and physiology. That's just one example, but, you know, cell wall physiology too, right? There's a lot of cellular level traits in building materials for the cell wall that's not leveraged, I think. And that, you know, I think there's a lot of biomaterials and bioproducts fields that could really take advantage of single cell technologies. And I think some of the stuff that you know, EMSL can do could be really interesting if applied to you know, these cellular traits.
Yeah, that's great. And I think that's kind of, to me, that can be related to, you know, you listed the technologies that actually required by Plant Cell Atlas. And I'm sure EMSL could contribute very effectively, at least in some of those. And I think we probably need to find some ways on how to actually do this and find some, you know, infrastructure and fundamental ways of putting together specific projects that comes from different labs, including EMSL, and shape it in a united form of Plant Cell Atlas. I think that's something that needs to be discussed more. For example, if EMSL wants to contribute in single cell proteomics or multi-imaging of data integration, how those funds can come from, how those specific projects can be defined, and then how all of this can be integrated into actual Plant Cell Atlas, working with this really diverse community, because this is really diverse from different parts of the world. So I'm sure we'll learn more about how EMSL can contribute in our one-on-one meetings today and Thursday, and hopefully we can establish this and use this opportunity to initiate some more productive talks and collaborations in near future to move forward.
Yeah, that would be great.
Okay, great. If there's no more questions, I want to thank Sue one more time. That was a great talk, great discussion, and hopefully that actually initiates some more discussions.
Okay, great. Thank you, Amir.
Thank you
everybody.
so much.
Bye-bye.
Thanks, Sue.
Bye.